Good morning for colleagues in the United States, North America, and good afternoon for colleagues in Europe, and good evening for colleagues in the Middle East, Far East, and Near East. It's a privilege and honor to be here today in this distinguished webinar hosted by Sheikh Hamdan bin Rashid Al Maktoum Award for Medical Research. Maybe before we start the insightful presentation from our colleague, the eminent speaker, Professor MAGID Abou-GHARBIA from Moulder Drug Discovery Center, I will be briefly giving you an information about Sheikh Hamdan bin Rashid Award for Medical Sciences. So this novel cause medical award has been established in 1999 by the visionary of the late Sheikh Hamdan bin Rashid Al Maktoum, who has been the engine powering and the driving force to support medical sector at all levels, whether this is research or healthcare service providers. The Hamdan bin Rashid Al Maktoum Award over the years has established outstanding partnerships with many academic institutions, healthcare providers, supported so many eminent scientists at the national and international level. These efforts should not be left unnoticed, and there are many outstanding outcomes of people who have been awarded this distinguished award. To mention a few of them, 2 Nobel Prize winners in physiology in 2010 and 2020, Sir Robert Edwards and Dr. HARVEY J ALTER. These were 2 previous winners of the Hamdan bin Rashid Al Maktoum Medical Award. I would also like to here at this level would like to welcome His Excellency Mr. Mirza Al Sheikh and all members of the Board of Trustees of the award for the great support for the award at all levels. Also, I would like at this stage to welcome our eminent speaker, distinguished speaker, who has been and continue to be a role model for people who are working in the drug discovery field, Professor Dr. MAGID Abou-GHARBIA. Professor MAGID finished his undergraduate and master degree from Cairo University in Egypt, and then he moved to one of the Ivy League schools in the United States, the University of Pennsylvania and Philadelphia, and then he continued his career through the NIH postdoctoral fellowship for many years, and then moved to work as a scientist, vice president, and discovery leader at Wyeth Pharmaceutical Company. For people who doesn't know Wyeth was one of the giant pharmaceutical companies that then later was acquired by Pfizer. Then after, he moved to Temple University, where he was the founder of Moulder Center for Drug Discovery Research at Temple University, as well as a professor of medicinal chemistry at the College of Pharmacy at Temple University. To mention very few, because of the time limitations that we have, very few of the discoveries and achievements of Professor MAGID. He has on his shoulder 10 marketed drugs, 10 drugs that are in human consumption in the market right now. This is something unprecedented. It's something very unusual to achieve something like that, 10 drugs under the name of Professor Magid in the market. He has more than 121 publications in top journals, more than 125 US patents, and more than 350 patent applications and international patents, and so on and so forth. It gives me a privilege and honor to present Professor Magid, who is going to give us insightful talk that is going to be about phenotypic and target-based drug discovery approaches for the discovery of innovation or innovative therapeutics. So Professor Magid, my colleague, the floor is yours.
Thank you very much, Professor Talib, and thank you for the Hamdan Award organization. It's an honor and privilege to be the opener of your webinar series. And I'm glad to share with colleagues some of the effort done in drug discovery. So as Dr. Talib mentioned, we'll talk about the approach for drug discovery, and I'll take you for a brief journey. Okay, I hope. Brief journey today to talk about the two approaches. What is the phenotypic and target-based, and what the attribute of these, and then example of some drug discovered from pharmaceutical company and from academia, and some concluding remark. Let me say that actually there are two approaches to discover drug, and the one which we hear called phenotypic actually is called compound first. What people mean by that you have a series of compounds and you're going to look for activity of this compound through some of the available screen. This screen could be coming from cell, coming from diseases or animal model and so on. Then after you find actually the activity, you try to find the mechanism of action and you move them forward. Unlike this approach, the compound first approach, there are mechanism first approach, and this is referred to as target-based drug discovery, where you have the disease and you try to look what the affected disease and what the mechanism where you can actually modify by compounds and you move them forward. So that's the mechanism first, or some people refer to it as reverse pharmacology. And these two approaches, you can reach your goal of getting your drug candidate. Either approach, you have to go through the process, and here I summarize it for you, where you have to know about the disease and what really modulates the disease, the test, and so on. But with phenotypic, it's compound first and mechanism later. With the target-based, it's mechanism or target first, then drug discovery later. And they have advantage, both of them. Of course, when you start with compound, then you reach a drug faster, and that's what we call first in class, because you came up with the drug, but that drug may not be optimal drug. So then when you do the mechanism of, and then you study the drug, you come up with what we call best in class or follow-on. Sometimes they both overlap, and your first is your best of class. And we'll give example of that. Of course, through the years, and we know how we discovered penicillin. It came actually by chance from phenotypic screen for infectious disease, and that's how we were able to come up with the first antibiotic in that time. Then later on in the 80s and the 90s and so on, with the deciphering of the human genome, then the people start focusing on targets and mechanisms. And this is how the process evolved in the last few decades. Let me take you now to a few examples because our time is short, and I will focus on a natural product and then CNS drugs. And then we go into an academic example. So for natural product, as we know, it's provide a very fertile source for drugs. but really how you approach natural product is important. You can get drug from natural product using one of four approaches. Use the natural product as itself, and that's the natural product drugs, or you modify the natural product and do semi-synthetic, then use it as drug, or you link your natural product through a linker, and that's the antibody natural product conjugate, or you can feed the microorganism and get your natural product with some activity. All of these been applied successfully, and there are drugs in the market from these actually approaches. I'm going to mention one or two today. So let me give you a start here with something called rapamycin. And that came from soil sample, actually came from the South Pacific. And usually the scientists, they take these soil sample and do fermentation and isolation, and they came up with that structure on the right. It's very complex. Look, 31 membered ring, and it has so many chiral centers. At that time, they were doing phenotypic screen, and they found it has antifungal activity. So that was great. They got the soil sample, then they got something to use an antifungal. It went right away for development. Unfortunately, it failed. It didn't really do well. So they actually, while doing a phenotypic screen, they realized it has immune suppressant activity by testing that phenotypically, and that was very important data. So at the time, the researcher and the organization decided, well, how about transplantation? Since it's immune suppression, if we transplant organ, that will be great. They tried this, and indeed, it increased the survival rate of the graft organs, and they compared that with cyclosporin here, shown in combination in red, and with the drug itself in blue. And that was actually very successful. Then they tried later, and they figured, well, how about if you use the drug by itself? And it was more potent, actually, for increasing the survival rate. But one good observation, it was very good for enhancing the kidney function. And that actually was the key for the company to say, that's an immune suppressant for transplantation, but also this drug is mainly enhance the function. And that came as Rapamune, discovered in the market for transplantation, and was extended after that for some rare lung diseases. So that actually shows you the phenotypic approach. But that time, when we did the compound, people start looking for mechanisms. And that came in the mid-90s, and they found there are two proteins. I highlight them in both on the right here, called the mTOR, which is mammalian target of rapamycin, and the FK binding protein. And so these actually two proteins were confirmed back in the mid-90s by crystal structure with actually rapamycin with these protein domain. And that opened a lot of good indication. So people said, since they're working downstream, they could be working for cancer, or they could be neuroprotectant for stroke, or to prevent damage in the brain for multiple sclerosis. So all these opportunities were approached. How you approach this opportunity if you are a scientist? As I showed you, you can either use the drug itself, and they did that for transplantation, or you can make analog, and they call it rapalogs, which is really semi-synthetic compound to work with. So here is what they do. They said the molecule, for people of the non-chemist, I highlighted in color, have several regions where the chemist can involve its approach and make changes for these. But for my team, we decided to stick with two parts of the molecule, the 42-hydroxyl and the triene in the bottom. The reason we picked that, because they were far from the effective protein, and we figured maybe we can come up with new drugs with these two. So the scientists went and made a lot of changes. For the non-chemist, these structure represent ester, representing ether, representing so many. They made about 700 compounds, and they file over 100 patents in these. So what they did, they tried to explore now the mechanism for this protein for cancer as anti-proliferative. And the scientist who did the molecule, I highlighted in red, he made simple changes in 3-step synthesis, and that actually called hindered ester. And that compound was, for the terminology, they call it CCI, cell cycle inhibitor, and that compound found to have anti-tumor activity. For the pharmacologist and the researcher, yes, it was tested in vitro in several cell lines. I highlighted here the breast, prostate, and ovarian, and you can see good activity with low IC50. Also, the compound was for the brain human glioma, was very good, effective in working in dose-dependent fashion. And that encouraged the company to move it forward to the clinic. And this is the compound which they call it at the time Toricil, and against interferon. And look at the green curve, which is show the survival rate increased by 3 months. And I want to give you a secret, or not secret, it's fact. When you work in research, if you increase survival rate by 3 months, FDA will love you and hug you and give you approval right away, and they call it priority approval. Any drugs in cancer increase survival rate by 3 months and more will get priority approval. That's why if you check all pharmaceutical company, close your eye and say they're working in cancer. Yes, because it's easy to develop drug. And that's what happened to Toricil, was approved in the market, and actually for renal cell carcinoma, and actually now they're examining it, Pfizer, for other uses. So I showed you in simplistic way that you start with phenotypic, where you have the rapamune or the rapamycin in the alcohol. Then you make different changes, ester, ether, triene. Then a lot of drug came out 10 years after the rapamune came in the market. And these drugs, we call them follow-on immunophilins. And they have very good activities, including the triene, as I told you, and that's for stroke in the clinic by Pfizer. Let's take you for CNS disorder. I want to tell you, CNS is not an easy topic to discover drug, and the success rate is low, and target-based, it doesn't work that well because the complex nature of the mechanism and the gene involvement and these multi-symptoms for diseases. So your answer here will be phenotypic screen, and that's where we discussed as antidepressant effects. We counted on the use for serotonin, norepinephrine, and we said we'll do screening. And we focused on that serotonin, norepinephrine, because anyone who have diminished serotonin, norepinephrine in the brain will be depressed. Anyone who have more dopamine will have psychotic episode. So these are neurotransmitters are used, phenotypic screen, to come up with drugs. And as I showed you here, this is the showed you the presynapse and the synapse, and where the norepinephrine and serotonin come into synaptic cliff. If you have reduced serotonin, norepinephrine, people become depressed. For us as a scientist, we said, how we do phenotypic screen? We said, well, we saw some company working on some activity, and we have molecule in the company for analgesic, which failed miserably, and that molecule is shown here, called ceremadol. So I discussed that with the leadership, and I said, we can use this in our phenotypic starting points, and we can do actually what the advantage and disadvantage of the molecule. The disadvantage in front of you in the red, the molecule is chiral, so it doesn't exist as one molecule, and it's analgesic. We have to get rid of both of these. Yes, the chemist did minor change, and he removed the chirality, only 1 out of 2, and he lost the analgesic. How we put the antidepressant? We know from the literature that gamma-fixin has depression, and it has this small, call it alkylamine. So let's put alkylamine in our molecule. We did, and it brought antidepressant activity, but weak. Then the scientists took that and optimized, and when they optimized, they put the methoxy after they made several analog, and that actually came out of 6770 compound, which was antidepressant activity. Indeed, that molecule gave us what we look for, a reuptake inhibitor. That means the serotonin norepinephrine released, instead of being reuptake back, it will stay longer and will treat the depressed patient. And that was a good thing for us. And we moved in the clinical trial, and that brought Effexor as first serotonin norepinephrine reuptake inhibitor called SNRI, and was treating a lot of patients, over 20 million plus, and the peak sale was 4.5 billion. Companies approach the same thing as we did, and we call it the Me Too. Actually, most of the company tried to imitate, and they went to try to make a follow-on. This took 10 years from the company to follow on Effexor, and I showed you the list, including the first one, which came also from my group, and this is, I call it son of Effexor, called Prestige. And the only difference, does not have the methyl group, and it have the hydroxy, and that's also been sold on the market as Prestige. Then Lilly and other company have these drugs. They were not as effective. Cymbalta, some of you who know pharmacology knows that it's not really antidepressant, it's more for pain treatment. So Effexor remain first and best in class of the SNRI antidepressant. And that's actually was good to show you how some of these phenotypic screen been approached. And now I'm going to take you to example in academia, where actually in another lecture, I would tell you that drugs being discovered by industry, by government, by academia, even though the majority by pharmaceutical industry, but academia play also an important role, especially in the recent decades. And let's talk about here what we have in the Department of Pharmaceutical Science and School of Pharmacy. We have several projects ongoing, and I highlighted for you on the top line, those are CNS projects where we're working on cocaine addiction using glutamate transporter, or 5-lipoxygenase inhibitor for Alzheimer, 5-HT7 antagonist for inflammatory bowel disease. In the lower, there is other approaches, but two of them for cancer, CDK9, RAD52, and other for viral projects and diabetes. And because of the timing, I'm going to just take one project, which is innovative projects started here at the School of Pharmacy, which is CDK9 inhibitor. Let me tell you, when you look at the epigenetic drug as anti-cancer, and this is actually was new trend back in the early 2000, there weren't really too many epigenetic drugs moving into the clinic. In the year 2000, started with the first generation, which is the DNA methyltransferase, DNMT, and then followed by the DNMT and HDAC, which is histone deacetylase, and then third generation immune therapy, and fourth, and so on. Then now there is over 30 drugs in the clinic. What's been discovered in the market, the first one came, the first one came was Veronostat, which is actually HDAC inhibitor, known as SAHA. My friend, the late Ram Breslow, actually team discovered that and went to the market 2006. Now there is other compound came also on the market. And I want to add to point here, my colleagues, the moderator, Dr. Akshat Talib and I collaborated on HDAC, and we just published recent paper in that projects. So now there is a lot of drugs in the clinic, but none of these drugs are targeted the CDK's actually approach. So let me just mention to you that these cyclin-dependent kinases are three kinds. You have regulator of cell cycles as cancer and as cytotoxic, and you have regulator of transcription, and these involve in epigenetic research. And the third class is really unknown. It has several CDKs, but the role is not clear. So for those involved in epigenetic, are there epigenetic modulators? Can you have drugs in these? And then I'll give you the answer in the next few slides. The answer, hopefully, yes, because we published that 2 years ago, that we target CDK9 to reactivate the epigenetic silence gene for cancer, so they can be good anti-cancer agent. And we actually came up with a good inhibitor which showed in vitro and in vivo activity, and it sensitized the cancer checkpoint, so it can be also used in combination. And that started with phenotypic approach. We did screening of 3,000 natural products here at the Pharmaceutical Science Department in School of Pharmacy, and we tried to look for lead compound. Indeed, we have a hit, and this first hit I highlighted for you. The important is called aminothiazole, and then we modified it and we came with non-selective CDK inhibitor, and then we further optimized and we came with MC295. And that compound came from out of 100 compound made as CDK inhibitor. And I want to say CDK inhibitor because we looked into that, and indeed, among the different kinases, was more selective for CDK9, and it showed more selectivity against other. We also did the human genome screen, was mainly for kinase, a little bit with ST family, which involved in MAP kinase, also another anti-cancer, so it will be a plus, but mainly CDK9 inhibitor. And we actually said, is it CDK selective, CDK9? And we did some computing with the binding site of different CDK, and indeed, that actually bind to the binding site of CDK, and more so because of the nitro group, which do hydrogen bonding in the CDK9, and that actually prove our point. So how about if this molecule have good anti-cancer activity? We looked at the in vitro for their anti-proliferative and colony formation, and we did test that against the first lead HH1. And you can see in the blue here with the MC was more potent, almost 20 to 50 times more potent than the early lead HH1. And we took it into in vivo because that's the golden standard where you look for in vivo with transgenic mice. And you need to look at the tumor size, survival rate, and any toxicity. And yes, for the transgenic mice, after we induce the tumor, and we but give them the MC285, the compound, I'm sorry, 295, there is a mistake here. That 295 reduced the tumor size. And here the 295 enhanced the survival rate, and there is no weight loss. If you see the green curve and the blue curve. So that's actually fulfill our criteria that you come up with epigenetic modulator as anti-cancer, which active in vivo and prolonged survival rate, and there is no actually any adverse effect. So what we did, we went from the phenotypic screen, we got the early lead, we looked at the mechanism later, and we found that CDK9. Then we said, okay, in vivo and in vitro was good, and the optimization gave us 295. Then we moved it to development. And that compound here, I summarized the feature here, which I just mentioned to you with its in vivo activity. And in the School of Pharmacy, like other universities, they go for entrepreneurial. So they advance for development, and they got a company called Epigen Onco, which moving that forward for development. So let me just give you some concluding remarks so we can have good discussion. In summary, we have the phenotypic screen, and it has some limitation because of the low actually throughput. And that's why people turn into target-based. But believe it or not, now there is back to phenotypic screen because fewer first-in-class drugs is being discovered. If you look at the FDA approval, not too many first-in-class. So now we need to discover more first-in-class drug therapeutics. So for you as a scientist on the bench, when you work in drug discovery, you have the two approach, phenotypic drug discovery and target-based. And I showed you that you do the mechanism later in phenotypic, you do it earlier in target-based. Are they competitive? Do you pick one over the other? My answer is no. They are synergistic. And when you combine them together in your research, you can increase your success rate. And that's indeed what we did. And I gave you a few examples. And this example summarized here, 2 from pharma, 1 from academia. And they are actually first in class, discovered from Phenotypic, and follow on and could be best in class later on after the patient used these drugs. I summarized that in a publication end of last year for those who are interested, where we talk about resurrection of phenotypic screen. And you look at the lower compound, I discussed that, but I didn't discuss the other 2. And as I said, this is phenotypic screen rising from the dead, and we call it the resurrection. And we were laughing, my collaborator and I, when we call it Lazarus come forth. But let me say, we couldn't do this without talented scientists. And Dr. Talib said, I was fortunate to work with very talented scientists, people from Wyeth. They were really very dedicated. As he mentioned, discovered several drugs. Many of them are first in class, and they have 4 blockbuster compounds, which won the Heroes of Chemistry Award in the ACS Society. And here in the Department of Farm Science, the Moulder Center, and they are dedicated scientists. They have over 30 patents, and there are 3 spin-out companies so far and 2 in progress. Several collaborators, and more important, we have visiting scholars come here. Some of them came from the Middle East, and some came from Algeria, and some came from Europe, from different countries, from Barcelona and others. And we open our lab for training. And I wanted to just appreciate their input. And I will close here. And I hope I didn't go over my time. Leave it all to you.
Thank you. Thank you very much, Professor Magid. That was really very insightful presentation. Outstanding discoveries, actually few of them only. I know how many hundreds of scientists were trained under your mentorship, Professor Magid. So you not only enriched the market with first-in-class drugs, but also empowered the market with great scientists from diverse backgrounds, as you mentioned. So thank you very much. And I think we will leave the questions and answers after the next session. So please, for the audience, if you have any question, please don't hesitate to send it through the chat link on the screen. We will be hopefully discussing a few of these questions. But now we will continue this webinar. And at this stage, I'm privileged to introduce Mrs. Athira Sujathan, a postgraduate student to represent the young researchers. Mrs. Athira has completed her master's degree in pharmaceutical chemistry from Amrita School of Pharmacy in Kerala, India. So the virtual screen is yours, Mrs. Athira, please.
Thank you, Professor. Good morning and good evening to everyone here. I'd like to introduce myself. Myself, Athira Naya, a postgraduate student from Amrita School of Pharmacy, Amrita Vishwavidyapeetham, Kerala, India. It is an honor to be a part of this webinar with an eminent personality as the speaker, Professor MAGID ABOU-GHARBIA, and another great personality as the chairperson, Professor Talib Al-Tal. I take this opportunity to thank Mr. Abdullah bin Saukat, Executive Director of Sheikh Hamdan Award for Medical Sciences, for giving me such an opportunity. Before I begin, I would like to just introduce a little bit about my university. As you all know, Amrita Vishwavidyapeetham is a NAAC-accredited A-grade multi-campus, multidisciplinary research institution with 6 campuses, 15 constituent schools in 4 states of India: Tamil Nadu, Kerala, Karnataka, and Delhi. It offers a total of 207 undergraduate, postgraduate, integrated, as well as doctoral degrees. Now, today I have been assigned to give a few words about academic researchers, the challenges faced by the academic researchers in today's world. As we all know, academic research is all-encompassing and covers a wide range of research areas and investigates various aspects of target. The early discovery process in majority of the academic programs is initiated with target identification, validation, development, optimization, and much more. After the target identification, it is followed by discovery of lead molecule, which is in further taken for in vitro, in vivo, and even clinical trials of compounds. Now, as a student, few of the challenges, not everything, but few of the challenges faced by me are mainly the time and cost. Since academic drug discovery is a time and cost consuming process, many of the students do not move forward for further education. Either they stop it with their undergraduate or maybe postgraduate. Further, we can see is that even if you move forward, grants and funding, mainly in the case of drug discovery based on software, that is the, as Professor has mentioned, target-based drug discovery, phenotypic, and much more like fragment-based drug discovery. Everything involves software-based work. So the software accessibility and the grants and funding being given to the students are much less when compared to the normal grants provided. Another issue we have seen is that animal models mostly cannot recapitulate the entire disease, what we are supposed to do research on. At the same time, even unknown pathophysiologies of many disorders has made target identification challenging, much more challenging. I have many more to say, but I would like to conclude with a small question that how can we rectify these challenges, or what is the future of drug discovery? Over to you, Professor Magid and Professor Talib.
Yeah, thank you so much, Athira. Thank you. Insightful thoughts, really. So I think we are ready to receive questions and to open the floor for questions from the audience. I can't really see any question in the chat. So Professor Magid, can we open the discussion among us
Sure.
At this stage?
Sure, until we have a question, yes. Well,
Yes.
Athira, let me tell you, usually when I give lectures, I make sometime lecture between the comparison of pharmaceutical drug discovery and academic. And there are several factors. Of course, when you look at the academic, when you talk about resources and so on, they are not really like the pharma, but also don't forget pharma is a business and they really govern by projects which are going to bring revenue. Unlike us in academia, we can work on rare disease, we can work on others and so on. So, and there is one thing which everybody has to understand. You cannot take the compounds or drug discovery from A to Z to the market. You have to partner with pharma. So my suggestion is always that you do the key studies and projects, because sometimes pharma, they have limited programs, and when they see what advances you have or some of what you refer to, you have some lead molecules, which is actually need to be moving forward, but it's going to cost very expensive to do what we call enabling IND study, investigational new drug studies. These are very costly. So some of the company will say, okay, we can partner with you and see maybe later we can license this from you and the university and so on. So definitely you won't have the resources, but you can do the area of expertise we have, where for our scientists, you have strong medchem and pharmacology and related. We have in our school, I'm sure you have in yours, you have the animal colony and you have other things. Here we have proteomic group at the School of Pharmacy and others. So you can utilize some of these. We have high throughput screening engagement. We actually instituted here something which most of, not all of the university don't have. It's called in vitro ADME. We do this high throughput screening. If you have any molecule, you want to know if you're making drug of the brain, is it reach the brain or the brain barrier will stop it? We can tell you that. So we do this in grants with other collaborators, and sometimes we do this type of research. So you can get always the collaborative research from each other.
Yes. And if I would like to comment on what Professor Magid mentioned, Athira, you know that the drug discovery process takes from 7 to 10 years on average, and it costs from 800 million to 1.5 billion US dollar to bring a molecule from the organic chemistry shelf to the market. So it's extremely lengthy process, extremely very costly process. And the attrition rate is extremely very high. The failure rate is extremely very high in drug discovery. Now, to add to what Professor Magid mentioned, it's unfortunate to see that in the developing countries like India, the Middle East, and many other countries in the Near East, the story of drug discovery is completely different to the story of drug discovery in North America, in particular in the United States. I visited Moulder Drug Center in 2018. I visited Professor Magid. I presented at his place. Now, surrounding the Moulder Drug Discovery Center where Professor Magid is working, there are hundreds of pharmaceutical companies across the road from where Professor Magid is working. So the partnership between industry, academia in North America is very tangible. It's huge. It's unfortunate to see that it does not exist here. And this does not mean that we should be frustrated. No. Remember that the discovery of drugs starts in the medicinal chemistry lab, right? And this is actually one of the most important milestones in the drug discovery process.
Yes, I think actually towards the 2020 now, mostly the first preliminary stage is being taken with software-based drug discovery. And I think many of them have a small question or a query that there are different algorithms for different drug discovery softwares. So which is more reliable, even if it is in a preliminary stage for post-graduation or maybe undergraduate course, which is more reliable among the softwares? Or maybe why is it that two different softwares show two different results based on different algorithms?
Well, let me just say, sometimes we have some of these we get some support or guidelines and so on. Same as you hear, many of you, about the rule of 5, and if you don't have these molecular weight and things that through your compound is not, you take these as guideline, but not actually as specific to guide your research. Because when people say you shouldn't have molecule more than 5 nitrogen, that's not true. There are drugs in the market which have 6 or 7 nitrogen. When they say molecules which 500 molecular weight, well, I showed you rapamycin, which is much more than that. So same with the software, I would just put these things into your sort of research and see you can get the proof from the lab work. If you do molecule which showed you that's going to be inactive, then you do the cell line and you found it's actually active in the cell line. So sometimes the prediction is not absolute, but it's very helpful to get that.
Yes, thank you, Professor Magid. I think there are a couple of questions, if you don't mind, Professor Magid. Sure. One of them is related to your CDK9 inhibitors. So the question is, did CDK9 inhibitors had no side effects?
Yeah, what I showed for what we did is we look at the side effect from some of the earlier projects. Anti-cancer was mainly affecting some of the, since you reduce your tumor, will impact also your weight. So we actually were monitoring that as one of the side effects, and some of the biochemical, which we also did, which didn't show any. But of course, you will get that more detail in the tox study for the enabling IND studies. For us as an academic institution, we can offer a few things where we can do some of the things effect from doing some biochemical or some of the weight, or if you have CNS effect, get some molecule, maybe hypnotic or lose conscious, or so these are, we call it side effect observable for us in academia, but you need to have detail in 2 species, and that's what you run. You run for 28 days, 2 species, and it costs you some money. And of course, some of our partner will do that.
Excellent, thank you. So another question from Hiba Sayed, Professor Magid. How do you recommend I proceed in my education or research work to work in drug design? What's your recommendation for Hiba, Professor Magid?
I think Hiba, you need in your study to get the good background. And as I advise some of our researchers and some of the school also students, and then the career will open for you. If you proceed research, you have research to start when you in your research, and you follow up after you get your PhD into postdoctoral research and others, and you can do that in academia. You can do it in industry. There are some industry now offering actually postdoctoral fellowship, same as the offer for the undergraduate students, some of the fellowship for pharmacy students and so on. And even some of our students, when they graduate, they don't want to work in drugstore. They don't want to be working in pharmacies. So they actually can either follow pharmaceutical company, not as a researcher, but could be in medical affairs, could be in others. So the future is ahead of you. And if you're interested in research, so you have to actually keep advancing. We all learn every day. When you finish, you learn from your postdoc, from the environment with a different research mentor and so on. So that's what I suggest. And you may want to follow academic, industrial, and other approaches.
Thank you, Prof. I think, Professor Magid, I need to ask a question, or let's open a dialogue for that.
Of course.
So thank you. So one time Carl Sagan said, somewhere something incredible is waiting to be known.
Yes.
Now, we have so many unknowns, Professor Magid. For many drugs we don't, in the market, like ibuprofen, we don't really understand how they work.
Right.
You spoke about the serotonin reuptake inhibitor.
Right.
You discovered this drug while at YF.
Yes.
Unfortunately, you cannot diagnose a person with depression, like what you do for diabetes type 2. There are no biomarkers for depression. You cannot take a blood sample from a patient and tell him you have schizophrenia. You cannot diagnose a person with schizophrenia. A person with Alzheimer's, you cannot tell him you have Alzheimer's without interviewing him. You cannot really have a blood test to say you have Alzheimer's. So there are so many unknowns in our life. Even in 1918, we had the Spanish flu. The Spanish flu killed around 50 million persons, right? 100 years later, we have the COVID-19. What's going on? What's wrong with us? Are we too slow, or we basically don't know anything? What do we need to do? What do you recommend the scientific community to do to overcome and unravel the so many questions that needs to be answered for the benefit of the humanity?
Well, I think you have to always know we all always in the learning process. And I will share to what you said. Yes, indeed, when you have drugs, we don't know, but that's why the term drug repurposing. So once you get to know about the mechanism or what the drug affect, there is now many drugs being used for different indication, even though they originally were not for that indication. And that's actually the whole market of drug repurposing. And the other things, yes, for CNS disorder, as I mentioned, that you don't have say, okay, he's depressed, he's this. And that's one of the things I refer to the advantage. Now, when you get into those, some of the images came in, and then you can look at some of the PET studies and others, and for also particularly for the amyloid, for Alzheimer's disease. So this can reflect a little bit the status on the brain. And if you have a drug, then you can see if there is any improvement and so on. But these are all opportunistic and also depend on the advances. And as far as somebody who's depressed, how I'm depressed, well, that's why the physician and the psychiatrist, they are the best one to diagnose, because not everybody, if you have one of your relative died or people who have pets, your dog, then you are very depressed. That's not a clinical depression. You don't need an antidepressant. So you need to be diagnosed as clinically depressed. And they have already criteria for that, the psychiatrists and so on. So yes, every day we discover new things. And as I mentioned to you, I mean, I didn't dwell on it, but in some of the rapamycin, I mentioned multiple sclerosis. Indeed, they were actually, one would have bought the company for business reason, they didn't go for it. But there are a lot of opportunities to have drug for MS and for others and so on.
Thank you so much. There is a question from Maria Al-Mansouri. So her question, Maria, regarding the human genome for certain cancers, can any clinical research as targeted therapy as prevention in strong family history of cancer? I really didn't understand the question. So which means, I think what she means, can we use the genome crackdown of a certain family to say these people potentially will develop cancer or not?
Absolutely. So let me give you an example here. And, before the year 2010, when women used to go and get some of their genome, particularly BRCA1 and 2 and all that, and that wasn't actually that fashionable. After 2010, the number more than doubled. And if some of you recall the reason, I mean, when I gave some lecture, I gave this as a quiz. I said, do you know why? Then I put the picture of Angelina Jolie. And I said, look, she actually found that her mom, her aunt, her thing, they all died of cancer. And she wanted, when she found that she had mutation in her gene, but she went to extreme. She did the mastectomy and all this operation. But now there are drugs where you can actually work against the BRCA mutation, HER2/neu, and all the others. So the answer is yes. Some of these gene biomarkers are going to be very helpful, and that's what we call the personalized medicine or precision medicine. And you'll find more drug goes to the clinic based on that.
Yes, thank you so much. Another question, maybe the last one, or maybe we can take another one, from Ima Pinajic. She is asking, in your opinion, Professor Magid, what are the obstacles for pharma companies to bring R&D into the developing countries?
Well, I want to just say, and I'm not going to defend the pharma or things, I think it's always usually our fault in a way that countries in the Middle East, and I'm one of them, we really need to encourage pharma industry and research based on research. Most, and I say it bluntly, people may argue with me, there is no pharmaceutical industry in the Middle East and many countries actually based on research. They all based on generic because of you get the return on investment faster and so on. So this is something some countries start doing it. And I know United Arab Emirates, you guys have the Medical Research Institute, and I know some other country in your neighbor and others start doing that, more biomedical research interest. We need that. And don't forget, as I said, pharma business is usually based on the return on investment. And I'm in Egypt, hepatitis C was a major problem. 20% people have it. And yet we don't have research in Egypt about that. and we waited until the Solvovapir and other company, they will do it for the hepatitis, mainly in Europe and US, genotype 2, not genotype 4. And if it works in your country, great, will be more money for them. So that's what I say, it's a business, and that's how the research goes in big pharma. But we should start also from home, focusing on that too. Did I lose you here?
I'm sorry, I was muted. So there is another question. I think from Hafsa Ambareen, or she's asking about the progress in precision medicine. How do you comment on that, Professor Magid?
It's actually, there's tremendous progress. It's still early. And don't forget, actually, these things, when you develop some of the CART and other related and things, very expensive. So early on, that's why some companies, when they come up with the drug, they may tell you that it costs you 200,000 or so, but they made a provision, say, if you're not treated when you take the first dose, then we'll refund or whatever it is. So I would say precision medicine and personalized medicine is advancing and is actually going to be the wave of the future. And if you look at cancer, the death rate was actually very high in the early several decades. And now when people get breast cancer, the survival rate is over maybe 80, 90%. And because of actually using the specific drug to treat the specific mutation. And who knows, in the future also, we'll have CRISPR-Cas, where you can cut the bad genes and you put the good genes and so on. The first clinical trial is being actually ongoing by researcher University of Pennsylvania and so on, but this is still a long way to go.
Yeah, actually it went into clinical trials, the gene editing and precision medicine.
Right.
Yes, absolutely. Thank you so much. There's another question, if you don't mind, Professor Magid, asking, do you think that, this is from Yusuf Abdul Razzaq. The question is, do you think that the most promising drug discovery is usually coming from natural resources, or like medicinal plants, or synthetic?
I think actually it's a combination of both. And if you look here in the history of natural product, in the old days of, I don't want to say maybe before the year 2000, all the company have natural product department. After year 2000, go check Google's and they find Pfizer phased out natural product, and every company I know at Wyeth, we phased ours, but I resisted until I left actually, and I've been always champion for natural product. The reason is that because the low throughput for natural product, in order to do the fermentation, isolation, and the deconvolution to come up with the structure, and now we have combinatorial chemistry and so on. But now people become clever in natural product. They have actually, they do actually the very fast in doing the isolation and the structure characterization. So it's now, it's being returned back. But the question is, use natural product as is. In pharmaceutical company, they won't. They use the active constituent in the natural product. And as I showed you, when you have your fermentation broth, we'll see like 6 peaks, anti-cancer. Then we go separate the active peaks. Then we identify the structure. Then we make the drug. And there are several drugs in the market came that way. So yes, there is room for both of them.
Yes. Actually, there was a report in the Journal of Natural Product maybe 3-4 years ago that almost 70 or 60% of the drugs in the market that they are either natural or semi-natural products. So a huge percentage of the medications, they are nature-inspired or natural products. The problem of the natural products in drug discovery, if I may be allowed to comment on that,
Sure.
It's somehow having so many problems. One of the problems is their diversity. They are not diverse in a way to have a concrete structure activity relationship on one side, but also their availability relative to the quantities needed for screening and maybe clinical or preclinical trials. That's why people are moving to something called complexity to diversity strategy, where they transform or make molecules that are inspired or similar to existing natural products to overcome the challenges arising from using natural products in drug discovery.
Correct.
Thank you. Thank you, Professor. This is Mr. Haider Hashim. Thank you, Dr. Magid. Which CAD software package you recommend for molecular modeling?
Well, you can use actually whatever available for you. I mean, some people, when we were learning, we use the Tripos, and there are so many other came out after that. And even though I'm not a modeler, but you have to collaborate with a good modeler where you can actually analyze whatever you get from your modeling to pursue your next iteration in your research.
Yeah, actually, Professor Magid, there are so many programs available for free. You can even download them, like Paymol or maybe other Discovery Studio, maybe part of it. Yeah, many others. I think there's not much. There's a question which is from Mrs. Maha Al-Rakan. She's asking a question which is not clear to me. Drug manufacturing versus natural substances and question mark. I don't really understand what she means unless she can elaborate further on this question right now. That's the only thing. Other than this, from my side, I can always be thankful to Professor Magid. He's always great support to every one of us, including myself. I had the privilege, and I continue to have the privilege, working under his mentorship and partnership with his labs, and we publish together, and hopefully we will continue to work in other fields. Thank you so much. Thank you for Sheikh Hamdan bin Rashid Al Maktoum Medical Award, and thank you, Athira. And Professor Magid, if you want to say the last word, and then maybe Athira.
Well, thank you very much, all. It's a pleasure to interact and work with you. And I'm here if you need anything. Shoot me an email or anything, and I'll be glad to give you any opinion or any advice. And wish you all well.
Thank you so much, Professor Magid. It was very insightful presentation and discussion as well. Thank you so much. Mrs. Athira?
I would like to, it was a wonderful webinar. It was a very good presentation, and even the Q&A session, it was very informative. I would like to thank Professor MAGID ABOU-GHARBIA as well as Professor Talib for your precious time you had given to us. And thank you for actually explaining each and every questions, whatever is being given here. Thanks a lot.
Yes, thank you so much. And we thank, as always, Sheikh Hamdan bin Rashid Al Maktoum Medical Award. I had the privilege as a director of the Research Institute for Medical and Health Sciences at the University of Sharjah for many years to supervise so many projects, outstanding projects funded from Sheikh Hamdan bin Rashid Al Maktoum Medical Award, so many of them. They are always, supporting, tangible, impactful research projects at the University of Sharjah and even other universities in the United Arab Emirates. Thank you so much, and I wish you all the best.
Thank you all.
Thank you, Professor Magid. Have a good day, and Athira, take good care. Bye. Thank you.
Take care, everyone.